Population genetics has proven useful in modern science, helping to identify susceptible groups and prevent disease. In Africa and the Mediterranean, the relation between the presence of the sickle cell gene and the survival of malaria has been able to be explained using population genetics. So what actually is population genetics? Population genetics is the study of changes in allele proportions and the corresponding evolutionary parameters, including genetic drift, mutation, selection, migration, and mating preference. In our study, we set out to examine how selection affects allele proportions. We hypothesized that in a population that adheres to Hardy-Weinberg principles, except where the genotype relative fitness of p² is less than 1, then the p allele proportion in future generations will be significantly lower than Hardy-Weinberg equilibrium's expectation. Using the Fishbowl PopGen program, we completed a series of 10 experiments with a p² relative fitness of 1, which is our control group, and 4 experimental groups with p² relative fitnesses of 0.7, 0.5, 0.3, and 0. All other parameters remain constant. We then averaged 5 repetitive experiments at each relative fitness and used the chi-squared test to examine whether they were in Hardy-Weinberg equilibrium. Our findings showed that all experimental groups were not in Hardy-Weinberg equilibrium after the 10 generations. As seen in Figure 2, all the different fitnesses showed a significant deviation from the control group, suggesting that a decrease in fitness will result in lower allele frequencies and will continue to drop as the fitness decreases. Thus, our findings supported our hypothesis. From this, we concluded our results demonstrated directional selection, disfavoring the p² genotype. Our results mirror real-world application, where deleterious alleles with a lower relative fitness are removed from the population. Our findings also concur with previous studies, suggesting that selection can influence genetic frequency in a population.